So as you all know that using the PacBio HiFi long-read sequencing, we could resolve a lot of unresolved questions in disease genomes. So in this talk, I will focus on the bioinformatics analysis for the cancer and the rare inherited disease studies using HiFi sequencing technology. So so first I will talk about cancer study. PacBio has developed several pipelines for covering cancer genomic study as well as epigenomic, cancer genomics, epigenomics, and transcriptomic studies. So for example, we recently released a new WDL pipeline covering the somatic variance analysis using the tumor normal pair samples. And we also have the isoform sequencing analysis pipeline, which has already been integrated into our officially supported bioinformatics platform, which is called SMRT Link. Just give me a second. I want to enable the laser point. SMRT Link, which is targeting to the isoform construction. Basically give you the isoform level transcriptomic profiling, as well as also focusing on the novel isoform discovery. And we also have a standalone tool available on GitHub for the fusion gene detection. So just for your information. So in today's talk, I will mainly focus on our newly released HiFi-Somatic WDL pipeline, which is a cancer somatic pipelines. So here is an overview of the pipeline. As you can see that this pipeline actually covers the different types of variants, including the small variants, large-scale structural variants, and the large-scale copy number variations, and also cover the methylation analysis, for example, to detect the differentially methylated regions or DMR. And this pipeline can also offer the mutational signature decomposition as well as the HRD prediction. So actually, at the end of the especially for those non-bioinformatician users, that at the end of the pipeline, we generate interactive analysis report that include several key outputs of your samples. For example, the HRD prediction, the copy numbers, the SNV and the mutational signatures, as well as the final results of the SNV or variance detection, including the annotation. So, and as you can see that including the functional annotation. So after that, you could actually select your you actually can narrow down to narrow down to the variants you are in mind. You are interested in by searching the names or the gene names as well as other like their genomic regions like introns, promoters, and so on and so forth. Okay. So here's the details diagram of the whole workflow. As you can see that the workflow starts from the HiFi, basically our long-read sequencing, our long-read alignments, followed by a few basic QCs, for example, the depth metrics as well as the alignment QCs. And the pipeline offers, actually the pipeline provides the variance coding for different types of variants, including the small variants like SNV and indels using deep somatic and ClearS, and also the large-scale structural variants using one of the cutting-edge long-read somatic SV detection tools called Severus. So the small variants and large-scale structural variations will be further utilized for the homologous recombination deficiency prediction using chart. So, and as we all know that most of the cancer tumor samples, they are actually highly heterogeneous, right? So there are a lot of subclonal events, for example, the subclonal CNVs. So that's why in this tool we also recruit one of the tool from the Harvard Medical Foundation toolset called Purple for the subclonal COVID normal variation detection, as well as to estimate the tumor purity. Basically, the tumor purity is the proportion of the malignant cells or cancerous cells so harbored by your tumor samples. So as you might already know that, okay, along with our HiFi sequencing, PacBio HiFi sequencing actually offer the native of 5-MC methylation detection. So it means that you do not need a separate assay for 5-MC calling. So everything will be provided just by one assay over your HiFi WGS sequencing. So utilizing the native 5-MC information, we could also compare the methylation information between tumor and normal samples and conclude the differentially methylated regions in tumor samples. And we also offer the pipeline also provides the mutational signature decomposition derived from the SNV calling results. So as you know that after my co-Stratton lab published the first mutational signature paper in 2013, I think that will be after that the mutational signature decomposition has already become routinely practiced when people study the cancer whole genome sequencing where we have enough number of samples, where we have enough number of variants detected. So the mutational signature will help us to, as you might already know, help us to investigate the accumulation of the mutations and how those how those mutations will eventually lead to the oncogenesis events in the underneath cancer genomes. So eventually, all of the variants derived from this pipeline will be put into different annotations. So for example, we first will perform those straightforward and simple functional annotation using different tools for different variance types. For example, for small variants, we use VEP. For large scale structural variations, we use a tool called NOSV. But eventually, all of the variants will be put into the more detailed cancer genomics annotation. Using the database from the IntelGene, which is called CCG, Compendium of Cancer Genes, for better clear cancer genomics view of the variants we derived from this pipeline. Okay, so next I will quickly go through some technical implementation details of this workflow. So first of all, this workflow is written in WDL, one of the common workflow language. And since it's written in WDL, so it can be executed using two popular WDL engines, which one is MiniWidow, the other one is Cromwell. And of course, to increase the portability across different platforms or different servers that we actually recruit Singularity as a container image management system. So basically, we wrap up those third-party tools in the Singularity containers and then put them everywhere when you want to apply this pipeline. So and internally, this pipeline has been tested on. This pipeline can be run on any computing environment, including cloud. But for HPC, we prefer when you're dealing with a large-scale data set, we prefer to submit the jobs through any job scheduler, for example, Slurm or SGE or LSF. So basically, internally, we prefer MiniWidow plus Slurm workflow. And we provided a MiniWidow plus Slurm workflow on our GitHub page for customer to adopt. But of course, this pipeline can also be adopted on any cloud platform like AWS. Just for information. So the usage of the pipeline is quite simple, as there's only one entry point. So basically, you just need to put everything needed by this pipeline through a single input JSON file. For example, you just provided the information, the path of the tumor normal pair BAM files. And you also need to provide the reference files, which has already been uploaded to Genado. So you just download and unzip them, so everything will be ready to go. So after that, you can also tweak some task-specific parameters, but usually the default will be good enough for most of the users. So next, I will just quickly go through a few key outputs of this pipeline. So first, this pipeline provides the fully phased sequencing data in the haplotype BAM format, okay, for tumor and normal samples. And after that, we also the pipeline also provided the sequencing depth matrix derived from the MOSDEP tool, and a lot of alignment statistics. For example, the percentage of the primary alignment and the secondary alignments, and so on and so forth. And we also have the per alignment statistics, which is give you more details of your alignments. And for SV calling, basically the SV caller provided three different outputs. The first one is the raw VCF file for SV calling, which is annotated. So of course we want to basically push this a little bit forward, that after we remove those control basically the normal VCFs from the normal samples, we basically derive the somatic VCF variant set, which will be further annotated using SVPack for the simple function annotation. It's like something like here. And after that, we will further annotate this SV set using the cancer genomics database, for example. Just what I just mentioned, IntoGene CCG database. So in terms of small variance calling, we are similar to the SV. We also have the raw VCF files as well as the annotated VCF files using the VEP for function annotation, as well as the IntoGene CCG for cancer genomics annotation. And so basically utilizing the variance derived from the tumor samples, we actually trying to decompose the mutational signatures and give you some results and visualizations like this. So for differentially methylated regions, we provided three different output folders. One is the DMR readouts. This folder contains basically the raw DMR regions, which is annotated by in the TSV format. And we further annotate those DMRs basically by overlapping those DMR regions to the introns, promoters, or those important genomic regions. And based on those annotations, we actually further bind those DMRs into different categories. For example, for this TSV file, is harboring those DMRs overlapping with the regions 1 to 5 kb to the TSS regions, or those DMRs overlapping the 3', 5' UTR regions, exons, extrons, and some known promoter regions as well. So basically give you a more categorized view of your DMRs. And for CMV, we also have the CMV output from the HMF tools purple, and give you some visualizations, the circle plots like this. And we also I'm utilizing the SMV results derived from the ClearS. We also have and also the purity prediction results derived from purple. We also try to estimate the basically the major and minor allele frequencies using the CMV kit. So next, I want to share some of the benchmarking results for this workflow. So first of all, for SV coding, we are using some truth SV set derived from the melanoma cell line that we could actually using 60x tumor samples and 30x normal samples, we could achieve a very good recall, where 92% of the SVs from the true set can be recaptured. And actually, even using the 30x, even we basically cut down the tumor coverage to 30x, and we can achieve quite good the recall rate. Because of course there are some there are some drop on the performance, but there's mainly because of the number of reads covered supporting those regions. So there is a caveat I want to share with you. When you're doing the SV analysis using basically the long read sequencing data, there's one thing we need to take note that given the similar comparable coverage, that the short read sequencing data will have much more reads compared to the long reads because our read length read span is much longer, right? So that's why when we try those NGS tools for SV coding on our HiFi sequencing data set, we might want to tune down those basically the number of reads cut off for the NGS data might be too stringent for basically to detect the true variance using the NGS, using the HiFi sequencing data. So that's why we might want to tune down. So actually, if we tune down the minimum number of supporting reads to 2 for Severus and rerun the whole testing, that actually we can still achieve 92% of the recall rate for the true SV set. And as you can see that for out of the 57 true SV detected, we can actually cover some of the SVs with very low allele frequency, which is known to be challenging in the SV detection. Okay, for small variance coding, so for SNVs, we could easily achieve to over 98 over 80% of F1 score for the very low allele frequent SVs. And for indels, coding is a bit challenging, but we can still, using the deep somatic and 60x tumor samples, we can still achieve the over 60% of F1 score. And of course, for those more abundant indels, we can easily maintain over 80% of F1 score, or even 90% of S1 score if you're using deep somatic and 60x tumor samples. Okay, next is mutational signature. So we first of all we test the mutational signature decomposition algorithm in our pipeline and compare with the known signature derived from the short read data. Actually, we found they are highly concordant with each other. And one more thing I want to bring on the table is that although because we can only compare to the short read sequencing data, so actually I believe that because our HiFi sequencing can sequencing more challenging regions, for example those tandem repeats and segmental duplication regions, so on and so forth, so we could decompose. We actually can detect more variants SNVs, so we should be able to have a high resolution mutational signature decomposed in the real sequencing data set. Okay, next is for copy number variations. The result is similar that, okay, basically the copy number variation derived from our HiFi sequencing data using this pipeline is highly comparable with what we know from the short read sequencing data. And we actually use two tools. One is called CNV-Kit, the other one is Purple. As you can see that most of the major, basically the clonal events are actually identical between these two colors, but Purple actually can give you much higher, higher resolution of the SV CNV profiling, because the Purple can actually working on those subclonal CNV events. But that also means that when you're applying those conventional CNV coding tools like CNV-Kit, you might get some weird, non-integer CNVs. This likely is due to the subclonal events. Okay, so at the end of this session, I just want to share one example, which is showing the integral view which is showing that, okay, HiFi sequencing can provide the haplotype specific integral view of genomic and epigenomic profiles of underneath cancer genomes. So here in the melanoma cell line we tested, we could, within the PTEN tumor suppressor genes, we could detect, in tumor samples, we could detect 12 kb large-scale deletions, which might be missed in other technologies, okay? And then, and along with these 12 kb deletions, we could also identify, tumor-specific somatic hypermethylation within the PTEN promoter regions. Okay, these two events might be also associated with each other, right? So, and besides that, we also observe loss of heterozygosity events where, okay, this brown haplotype in normal samples are completely lost in tumor samples. So basically, all of those things can be done within just one assay. You don't need 2 or 3 assays to get this analysis done. Okay, so after that, I will move to rare inherited disease. As we all know that for the rare inherited disease, the haplotype resolved variance analysis is critical. Using PacBio HiFi sequencing, we actually can provide a full spectrum of variants, including germline variants, including those challenging ones. For example, the large-scale structural variants or complex structural variants and copy number variations, and also those variants falling into those dark regions or the complex genomic regions. For example, the tandem repeats regions and segmental duplication regions, and other dark regions. So at the end of the workflow, we actually can provide allele-specific, allele-resolved variance set for customer to use. So here's some a little bit more details that, okay, what kind of tools we have for rare inherited disease analysis using HiFi sequencing data. So here we actually recruited or developed by ourselves a few series of tools for the rare inherited disease study. So those tools marked as STAR is actually developed by our own selves. So as you can see that we use a deep variance, the Google deep variance for small variance detection, where using HiFi sequencing data, we can detect more 5% or more small variance due to our HiFi long read span, due to the long read span of our sequencing technology, which can extend into those challenging regions, right? So, and also our performance on those large-scale structural variations as well as copy number variations are much better, right? Comparing to other technologies with over more than 60% of genetic variants are detected. And we are also good at the genotyping in tandem repeats regions as well as the segmental duplication regions. And eventually, we also, dealing with, we can also utilizing those native 5MC methylation information come along with the HiFi sequencing to find those rare differentially methylated regions in cohort analysis, which is quite common in cancer, in rare inherited disease. Okay, next I want to talk more about the tandem repeats. So as you know that there's 4 repetitive DNA sequences in human genome, right? So some of them are, based on the pattern, they can be divided into tandem repeats and dispersed and other types of repeats. And for tandem repeats, we have actually the microsatellites and mini-satellites and microsatellites repeats. So among all of them, the mini-satellites are also called VNTRs, and for the microsatellites, they are called short tandem repeats, right? So they are known to be abundant in human genomes, and more importantly, they are more prone to have mutations. So they're actually 10 times higher mutation rate among other variants classes compared to these VNTRs and STR regions, right? So, and we also know that the over 50% of the SVs are actually derived from tandem repeats regions. So that's why it's important to study these regions in rare inherited disease. So to do that, we actually developed a tool for the targeted tandem repeats profiling, which we call tandem repeat genotyping tools, or TRGT or target. So this tool is starting from aligned HiFi reads, and which will be in which we will construct consensus alleles using the aligned HiFi reads, and those consensus alleles will be further annotated or using the well-maintained database, harboring those known tandem repeats patterns and the motifs and their expansion, so on and so forth. And if we are dealing with some disease models, right? So some of the tandem repeats that they are actually unknown. So for those unknown tandem repeats, we recruit the HMM model for the novel discovery of the tandem repeats. So and eventually all of the results will be visualized. Will be further visualized using a combined visualization tool called TRVZ. And more importantly, as we can also provide the 5MC methylation calling results along with the HiFi sequencing, but of course your protocol cannot include the PCR, right? So we actually have a CRISPR guided protocol for the tandem repeats profiling sequencing. So where we can actually keep the 5MC methylation information. So if that's the case, you can also look at the 5MC methylation information along with those tandem repeats you profiled. Actually, in the end of the visualization plot, you can see that you can actually decompose the the tandem repeats information into allele specific ones, right? So utilizing our HiFi long read sequencing technology. So the next thing I want to talk about is the segmental duplication regions, which is similar to the tandem repeats. These regions are also challenging because they are highly repetitive and they are dynamic. They're highly dynamic across different subpopulations, and more importantly, that they are known to be hotspots for structural variations, right? So that's why it's interesting to study them. And of course, they pose a lot of challenge to the read alignments and variance coding because they are highly repetitive regions. And in PacBio, we developed a tool called Paraphase. As the name said that, this tool is dealing with the paralog genes. So because a lot of the genes and the paralog genes, which might be associated with the rare inherited disease, might be falling into these segmental duplication regions, making the profiling of those genes and their paralogs very challenging. So that's why utilizing our HiFi long reads, highly accurate long read sequencing technology, that we could, so that we compose a strategy where we first harvest all the variants mapped to the genes and the paralogs falling into these segmental duplication regions, and after that remap all the reads to the genome interest. And after that, of course, the gene and their paralogs is not 100% identical, right? So there are some variants. So just utilizing those very, very rare variants harboring between the genes and the paralogs, we could further decompose the reads into different alleles. Some of the alleles might derive from the gene itself, and some of the alleles are actually derived from the paralog genes or the pseudogenes even. So then after that, we could, come up with allele-specific profiles of those challenging genes, the paralog genes falling into the segmental duplication regions. So here is the example that when we looking at the SMN1 and SMN2 paralog genes, which are known to be associated with the spinal muscular atrophy, which is a leading cause for infant death. So actually, for different patients, we could detect accurately different SMN1 and SMN2 allele-specific genomic profiles, just using HiFi sequencing and the Paraphase tool. So this will be very, very useful when we want to look at the genomic profiles and what is the disease-driven events in the analysis genomes. So another interesting topic in the rare inherited disease study is that people of course want to all of the variants to be fully phased, right? So because we want to look at the inheritance events that might be shared with the parental, might be derived from the parental alleles. So that's why we also developed a high phasing tool called HiFi. So this tool, comparing to the existing tool like WhatsApp, so there's a few advantages of this tool. The first is that it's not only phasing SNV and indels, right? So we want to fully utilize the large scale or more complex variants we derive from HiFi sequencing. So this tool actually can phase not only SNV and indels, but also SVs, the large scale structural variants, okay? And so, so here is just some benchmarking plots. So as you can see that compared to the existing WhatsApp, actually a HiFi has better performance in terms of the phasing blocks, basically the phasing N50, phasing block N50, and the number of phased genes, and also the number of phased variants, and of course, where WhatsApp cannot phase SVs, we can actually phase a lot of SVs, and we also suffer the lowest phasing errors. So at the end of this session, I just want to share with you that you don't have to run all the above tools one by one. We actually knitting them into a whole pipeline available on our PacBio WGS variance pipelines. This pipeline is not only available for HPCs, it's also available on different cloud computing platforms, including AWS. And if you want to has a more easy access to those pipelines, you could also access them through our tertiary analysis partners like GenX or Golden Helix. Okay. At the end of the talk, I want to share some more information about the population database we are working on, right, for HiFi long-range sequencing data set. So for those researchers who are working on the cancer genomics or rare inherited disease, they must know that those population-based variance database are quite important when we want to annotate our variants, right? So the variants derived from our pipelines. So that's why those population-based database like Genomeet'24 or Cosmic for cancer genomics, they are quite important. But now there's some gapping between the short-read sequencing data and the long-read sequencing data. Is that all of the mainstream population-based database, they are based on the NGS data, right? So based on short-read sequencing data. So actually, that's why we are leading the efforts to build a comparable database, population-based variance database, which are mainly derived from long-read sequencing technology. So that will give you a better annotation interpretation when you're dealing with the variant derived from the long-read sequencing technology like PacBio HiFi sequencing, right? So yeah, thank you. So this is the end of my talk. I'm willing to take any questions if there still have time.